All right. Thank you, everyone, for joining us, and welcome to the fifth installment of our Capillary LC series. This time, we're going to be taking a deep dive into detectors. Specifically, we're going to talk a little bit about mass spec and UV, but we're going to hear from Dr. Dasgupta, who is going to give us a great overview of Capillary LC as well as the associated detectors. And so, Doctor, would you like to take it away?
Thank you, Sam. It's a great opportunity for me to talk to you, although I can't see you. I am talking to my computer screen. But let me begin with perhaps 120 years ago, perhaps exactly today, Mikhail Svet was doing this in Warsaw. And if you go to the University of Warsaw, you can still see his statue. But, one of the things that Svet was separating chlorophyll and xanthophylls, basically leaf extract. And he saw all these colors separate, and he coined the term chromatography uh, from chroma and graphos. So it actually laid dormant for many decades before it was rediscovered and took off. Then actually took off much more in gas chromatography than liquid. Liquid chromatography will need another many years before it would also take off as ion exchange, amino acid analyzers, and result in more Nobel Prizes. So let me now say what constitutes capillary LC. The common size scales used in liquid chromatography are based on the inner diameters. The solvent velocities are typically, take it very approximately, 1 millimeter per second, regardless of scale, okay, in that order of magnitude. So when the diameter is more than 5 millimeter, we call it prep scale. The most common analytical scale today, which is nothing special, is 2 to 4.6 millimeters. And almost no man's land, it have to 2 millimeters. And then you again go to the capillary domain, which it's a very loose. Thermocols, they're 0.4 millimeter diameter ion exchange columns, capillary columns. Most people think capillaries begin smaller, probably 100, probably 300 micron, 250 micron. If you look at Axcend's recommended columns, all the columns are 150 to 300 micron in range. So there is a new thing that's emerging that people call nano columns, although it's still 100 micrometers in diameter, nothing nano about it, except the flow rates now are in nanoliters per minute. So the first capillary chromatogram these are packed capillaries. It came in 1978. Suda and Novotny. This is heritage. The founder of Axcend, Milton Lee, is actually a graduate student of Milos Novotny. And Suda was a postdoc. After he went back to Japan, he made his own name and made many contributions to capillary chromatography. So here, imagine almost 50 years ago, they're doing this. And this chromatogram looks pretty good until actually this took almost 80 minutes or 100 minutes really, to take place. And not surprising because the column is 30 the packing is 30 micrometer alumina that are not even spherical. The column was 29 meters long. Okay. So I'm going to briefly interlude into the so-called forbidden land, open tubular liquid chromatography. Why forbidden land? Because people have been trying to do this. It still has and show wonderful results, but it's still not commercial. But every detector that you can use in open tubular liquid chromatography, you can use that detector in any other kind of liquid chromatography. So if you can do this in OTLC, you can do this in others. But there are some things that's not quite true. Some things you can do in OTLC, you can't do in others. I will see that too. But the question that arises, almost all of gas chromatography is done today in open capillary columns. So why don't we do liquid chromatography in open capillary columns? The issue is that separation takes place on the wall. That means the molecule has to go to wall. There are no magnets that are drawing the molecule to the wall. Okay, so they can only diffuse there, and so that means two things matter: how far they have to diffuse and the diffusion coefficient. Can you induce some kind of a force for them to go there? People have tried that using planetary centrifuge in rotating columns at high speed so that they're thrown against the wall, but too complicated. So essentially, you have to rely on very small columns because in the liquid phase, the diffusion coefficient is 1,000 to 10,000 times smaller than in the gas phase. So the characteristic time depends on the square of the distance divided by the diffusion coefficient. So if you think about that diffusion coefficient and want to operate in the same time scale, your diameter is going to have to be, if the smallest diameter of or largest diameter of a gas chromatography capillary column is 600 micrometers, your capillary diameter should be square root of that. Okay, so that means that nothing much more than 25 micron is going to do very well. This is, you can also try to increase the diffusion coefficient. This is all this looks like a terrible chromatogram actually. And until I tell you, all these aromatics are being separated with water. Okay. And why that's important is because this is being done at 270 degrees C. Okay. So to keep quarter in the liquid phase, there's a lot of pressure, back pressure being applied. And under those conditions, everything is exiting as a gas. You can use standard GC detectors. These are actually uses a FID to do liquid chromatography. Okay. But it's very difficult. Water is extremely corrosive at high temperatures. And this is what happens even after saturating it with silica. A silica column basically is eaten in a few hours. It literally dissolves. So Nota first tried open tubular liquid chromatography. He he was very optimistic because even though he knew the theory and knew that it's going to require a lot smaller columns, he started with 260 micron columns, 5 meters long. He did not have detectors that will work with it, so he basically collected drops on a thin layer plate and used fluorescent analytes. But he did get separation, showed them, so that credit goes to him. And meanwhile, I told you about Suda, who worked with Novotny. And now, when Suda went back to Japan, he and his co-workers did much more work on OTLC. But really, the field was broken open by Jorgensen, who was also a student of Novotny and the academic granddad of our Sam Foster. The and So Jorgensen and Guthrie published this landmark paper in 1983, in which they show beautiful separations in open tubular format, and I'll show you some of them. And they conclude ultimately that the most uncertain area is detection. Here, we are farthest from our goal of realistic working systems, and any broadly significant development are probably many years ago. It's detection which will determine if liquid chromatography in open tubes, in capillary tubes, is going to be a real success. So here's some pictures from the Jorgensen-Guthrie's paper. This is 1983, okay? And they're showing on the top, you see basically coal tar extract. And on the left, you see a normal time scale, as they're trying to do it, what you can do in a normal time scale fast. And then if you go really slow, you can see all of those tiny peaks in there. They are demonstrating it, and this is all being done by fluorescence detection. And they're demonstrating it with small molecules on the bottom, where you see these fluorescent molecules being detected. They're using a very high power, not very high power, powerful mercury arc lamp and detecting the fluorescence. Nothing very fancy. So here I'm going to go all the way to ion chromatography, which is my favorite. This is one of our papers. And you can see ion exchange is actually not as efficient as standard reverse phase HPLC. So I think you will agree with me that this is a reasonably efficient chromatogram. In fact, it's better than what standard scale packed column chromatography can do today. This is a 25 micron open tubular column, requires only 50 psi pressure. That means operating at pneumatic pressure. Okay. And what is even more interesting that I'm going to have to move this somewhere. And what is more interesting is imagine that you have struck the column across your bench and you have the detector move along. And the detector then reads the conductivity and it generates this kind of chromatogram. Now imagine that you got this image, then you quickly brought the detector back and scanned it again and again and again and again, and you stitch these images together. So what are you going to see? You're going to see a movie of the chromatogram as the peaks are moving through the column, which we cannot see, but Sweat could see in his time. So here is exactly what I'm describing to you. Look at these peaks are going and separating along the way. Okay. Here is a much more powerful example. This is an 11 micron diameter column. And you will have to watch. I'll maybe show it twice, but you will have to see there are two peaks that will come out closer together, one taller than the other. First, one would be ahead, other will be behind. Then they will merge together. Then the one will take off. So you cannot see what happens within the column. So just because A comes out, the tortoise beats the hare at the finish didn't mean that tortoise was always ahead of the hare. Okay, so look at these two. By the time they merge, their positions have changed. Okay. So moving on. What more can you do with open tubular liquid chromatography? This is a liquid chromatogram. This is not capillary GC. Okay. This is done by one of my former students, his group, and 2,000 peaks in 3 hours, a pressure of 35 bars. Okay. This is E. coli tryptic digest, fluorescence tagged, laser-induced fluorescence detection. The column is 2 micron in diameter, and inside was coated with C18 functionality. Okay. And on the this is, if you push this in simple small molecule separations, these are 6 amino acids that are being separated under 1 second. Okay. And on the right hand side, you have this same device now connected to one of the high end mass specs at Pacific Northwest National Lab. And in this case, the flow is so small that you have to essentially dilute the flow, not with liquid, but gas-assisted introduction to the mass spectrometer. Okay, so there is nitrogen coming in through here and spraying it into the mass spectrometer. And so there are, it's amazing chromatograms that the peptide separations are from again, a digest from a single cell that can generate the complete proteomics. And as little as 75 picograms allowed them to completely identify 1100 peptides. So back to the real world, let's talk about detectors. So generally, you can divide it in multiple categories depending on your preference. There is direct contact, where however you're trying to measure, it's in contact with the solution, okay, or not, and that has the ramifications that whatever touching it might get tarnished over a period of time. And is it going to consume the analyte? If it's going to consume the analyte, then you may not be able to use other detectors after it, okay? And some will completely consume it. Some will consume it partially. You probably need to know how much it consumes before you make too much conclusion on the detector that comes after it, okay? Some detectors, these are not very easy to use in a capillary scale, are reaction-based, such as chemiluminescence. And then you have optical detection methods, absorbance, fluorescence, Raman detection methods, photothermal, which measures heat that develops out of absorbing light, and you have electrochemical detectors. There's a difference. People call conductivity and potentiometry, they put it under the same category as electrochemical detectors, but electrochemical detectors should strictly be where electrochemistry is happening. If chemistry is happening, then they're destructive. Okay? Conductivity or potentiometry does not destroy your sample. Okay? Strictly speaking, they're not electrochemical. They are electrical, but not electrochemical. Electrochemical methods right in the center are amperometry or voltammetry. They can be, if you carry out amperometry all the way to conclusion and eat up everything, then it's be coulometry. Coulometry is very difficult to do except in open tubular format, because all of it has to come to the electrode, and that means the distances have to be very small. Okay, so so detection differences between the scales is that everything you have, every point of connection, whether it's injector to the column to the detector, they are all sources of dispersion. In the macro scale, it's not so much of a problem. In the micro scale, it becomes increasingly more and more difficult. In many cases, you can't have a separate detector. You pretty much have to detect on the column, okay? So, or right as things exit out of the column. You cannot be connecting a separate detector because that connection will kill all the separation you've done, okay? Then there are other concerns like, yeah, so these guys did these wonderful things with a Orbitrap Lumos. It's about only about $1 million and plus some change. Okay, so if you have that kind of pocket change, you do it, but you're not going to carry it out to the field. Okay, so if depending on your need, you may be bringing in a cannon to kill a mosquito. Okay, then other thing is about scale, the mass sensitivity, how little you have. I'm talking all about picograms and so on and so forth. The are it always sounds very impressive, but often people aren't interested in mass detection limits. They're interested in concentration detection limits. One very some of you have heard of nanopore detections where the molecules pass through a very small pore and you can detect the passage of a single molecule quite easily, actually. What you don't realize always, they say, oh, I can detect a single molecule. So imagine you put a 1 milliliter solution containing a single molecule in the detector. How long will it take for that molecule to diffuse to that hole? So electrochemical detection method, very simple and very inexpensive. Basically, you apply a voltage to your working electrode and a current will flow between that electrode and a counter electrode. Generally, these electrodes tend to float in absolute potential. So you have to have some other electrode whose potential doesn't change. It's not always necessary, but typically we do, especially if you want to transfer a method to one lab to another, you got to have a reference electrode to make sure that it will work under the same way somewhere else. Okay. Here, clearly, the analyte is touching the electrode and electron transfer is occurring, redox processes are occurring. Okay. And so that means electrode fouling will occur, CRUD will form on the electrode. I'm sure some of you know that CRUD stands for chemically reactive unidentified derivative. Okay. So that means that you got to either replace the electrode or polish it or recalibrate it. It's really a hassle. Okay. So one of the things that Dennis Johnson came up with many years ago, but amperometry is a great way to detect many things, including carbohydrates, amino acids, and so on and so forth. So, but they all suffer from electrodes. Noble metal electrodes are used, gold or platinum, and they get fouled. Just because you're noble, you're not that noble. Okay. So pulsed amperometry was developed by Dennis Johnson, so within a sub-second cycle, the electrode is driven to, after the work is done at some modest oxidation potential, you drive the electrode to very high plus potential so that it'll surface, basically burn the gold surface to gold oxide, and then bring it all the way back down to reduce the gold oxide back to gold, and you start all over again. This works well, but still has a finite lifetime. Okay, when you do amperometry, so that you convert all of your analyte to the product, that is coulometry. Coulometry has the incredible advantage that it doesn't require calibration, because if you converted all of the analyte, you know how many electrons each analyte is going to consume. So all you do, current is number of electrons, what you're counting, then you know how many molecules you have oxidized or reduced. Okay. In potentiometry, I'm not going to go much into it. It's like a pH electrode. It has been used very successfully in early years. There are multiple papers in early 1980s where people are using 2 micron, 3 micron potentiometric electrodes. And imagine how long it takes them to put it at the mouth of the column, which is 10 micron in diameter. And Lord forbid anybody sneezes in that room. So, okay, so you can do extremely good stuff. I'm going all the way back to Jorgensen and Guthrie. It's a different paper now. They're doing this voltammetric detection on a graphite fiber electrode. Okay, they're analyzing human urine state, and here they're showing how you can differentiate between one analyte opposed to the other by applying different voltages. In amperometry, we hold the voltage constant. In voltammetry, we can vary the voltage either at different steps or we can scan during the run. So in this case, they're showing how that same chromatogram looks like once at 1 volt and once at 0.4 volts. So you can see that a lot more things and greater signals are obtained. Look at the scale also when you are driving it at higher potential. Okay, some of these peaks here, barely visible, will come out, and some of these peaks here are not only at all present, which are here. And then here is again a coulometry amperometry situation. They're going, here is the flow rate. This is percent of analyte that's converted. Notice that they're reaching nearly 100% conversion as you go slower and slower. The coulometric efficiency, you go to 95%. Most people will settle for 95% accuracy and never have to do calibration. So miniature scale, it works very well. So this has been done awful. And this is, you can do capillary electrophoresis, you can do microchip capillary electrophoresis. and detect, fortunately, many neurochemicals. Here they're showing dopamine and catecholamine. Here there are various biological thiols. This is a one of the common geometry in electrochemistry. Remember, the analyte has to come to the electrode. And how do you promote diffusion? You just spray the solution on top of the electrode. So basically, you have a wall, you have a jet hitting the wall. Right? That's a wall jet electrode. Typically, you can think of a disc and a tube positioned over the a pointed tapered tube on the disc, and so that's your wall jet geometry, okay? And these are some thiol separations. Conductivity. Conductivity is a bulk property, and basically the reciprocal of resistivity. So it don't seem that it is all that selective, but how can you detect? Because if a solution is electrolyte solution, everything conducts, how can you detect something in the middle of it? And also, if you typically separation of electrolytes involve other electrolytes, so the background conductivity can be high. So somehow you must reduce the background conductivity to reduce the noise to be able to successful. But what are the typical geometries? The typical geometry in the macro scale is basically two electrodes facing this way, and the solution perpendicularly flows through it. This is the same thing that is happening here, that you have a tube, except this is a capillary tube, and you inserted these electrodes in here. It's easy for me to say, not so easy to do when that thing is only 25 micron in diameter. And another is, here's a wall jet geometry. So here you have just a screw that can be adjusted to different distances, and you have a metal capillary ending, and you can vary the distance on them, and you can vary how close it comes. This is the exit view, and this would be the side view. You can have a sandwich electrode. It seems ideal, but very difficult to attain in practice. Imagine that you remember these telescopes, they have all this gold looking thing. So they're gold evaporated on kapton. Kapton is an insulator. Okay, so imagine you have gold layers on both sides of that kapton and you make a hole in it. Okay. So that the two sides are electrically isolated from each other because the kapton is in between. So you make those two the electrodes and pass the solution in between. Easy for me to say, but to connect to that hole is a whole different story. Okay? And there is another interesting geometry here is that imagine you have pair of wire, just like your lamp cords, okay? Except these are far, far smaller, and you have removed the coating from the outside, but they still have the insulation in between. Okay, so you take on these micro wires and you put it in at your capillary exit. So this solution will flow across those wires, and these are incredibly reproducible because those wires are coming from the factory with that separation, and you can do this very easily. Here is a portable IC that we made back in 1998. This is amazing to think it's 25 years ago. So here is this suitcase was custom made. Here is our pump. The cup is there, not part of the chromatography for my scale. Okay, but the detector, I already showed you the detection shell, what it looks like. But this is the kind of things you could generate. This is a gradient chromatogram, and it was a 180 micron column, if I recall correctly, packed column. And what else? I remember one thing is that I bought these whiskey flasks, and it took me a real trouble to get them reimbursed for our because the receipt said they're whiskey flasks. So now you have these contactless conductivity, they're commonly called, sometimes called C4D. And they're basically a tube with two rings on them. The rings are very close together, 3-4 millimeters apart maximum, often sub-millimeter apart. And what you do is you apply a relatively high frequency AC potential to one of the electrodes. The wall behaves as a capacitor, and that energy couples through the wall into the solution. Higher the frequency, the more effectively it couples to the solution. Then it travels to the solution only sub-millimeter distance to the other part of the wall when the other electrode is sitting, and travels through that capacitor wall again to the pickup electrode. Then you amplify, rectify, and have your signal. The whole idea is that the only thing that changes when you are detecting is the solution. So it's a solution resistance that controls how much current is flowing. So it's a conductivity detector. Actually, it's not, because the solution is not just a resistor, the solution is also a capacitor. And when you change the resistance of the solution, you also change the capacitance. Okay? Actually, you think about water, dielectric constant, look up the dielectric constant of sodium chloride solution. As you add sodium chloride, dielectric constant will decrease. So the capacitive current will fall as you increase the conductivity. So whether you see a positive signal or negative signal, at very low conductivities, you actually see a negative signal. Okay. Now, another thing is that you must have these electrodes are happy to, these currents are so small that these electrodes will talk to each other across the air. You think air is a complete insulator? No. When you're detecting picoamps of current, they're happy to talk to each other. So you got to have a metal foil that is grounded to keep them from talking to each other. Easy for one to say, but these are overall very easy to construct. They are very cheap. There are a number of sites, open design, open C4D sites, and you will find them easily. So admittance detection in the capillary scale. And here I am showing you both a macro and a micro example. This is a 5 micron capillary, and this is a 1 micron radius, 2 micron diameter capillary. We got some capillaries from Polymicro that were supposedly 1 micron in diameter. Polymicro gives you plus minus. It says the capillary diameter was 1 plus minus 1 micron. We we never could get flow through it. So I guess the part where it was 1 minus 1 micron prevented us. So here, this kind of a detector is simply attached to the back end of an IC in a split flow, and I'm looking at the bromide peak here. Here, I'm just amplifying a view of that bromide peak. This is the black is the blank. Here's a 50 nanomolar, that's injected concentration. By the time it comes out, it is diluted at least another 10 times. So this is about 5 nanomolar bromide that's detecting. Okay. And here are more examples of admittance detection. Lots of things you can do with it. It's an open source C4D, and there are lots of use in capillary electrophoresis. And now let's go to optical detection. When I have very little time left, but we talk, light has to be absorbed to do anything, and we talk about Beer's law all the time. I can't help it, but this is Pierre Bourget. He's the first one in 1729 said that light is extinguished exponentially with distance. Lambert, many years later in his book, put that in mathematical formalism. And all Beer did, another 100 years later, said, okay, the number of molecules in the path also extinguishes it exponentially. So that's the contribution of Beer to Bouguer-Lambert-Beer's law. So the moral of the story is, if you want to be famous, get a 4-letter word as your name. Okay? So Foster, too bad. Too late for you. At any rate, American Society of Metrology says this is what you should say, Bouguer-Lambert-Beer's law. So what that means, though, that the light attenuation is going to depend on the distance. And this is where it kills you when you are in small diameters. And the other thing that's misleads us in many ways is that we call it absorbance detection. But it's not really absorbance detection, because what you're measuring is the intensity of light that's coming. You're measuring transmittance. Then you're mathematically converting it to absorbance. Okay? The difference is real. Okay? The one thing in a perfectly designed detector, what you cannot overcome ultimately, you fix your thermal noise, Johnson noise, flow noise, you still have counting statistics, which is called shot noise. That means the uncertainty in the number of photon counts will always vary as the square root of the photon counts. So the signal to noise will always be n over square root of n or square root of n, square root of the number of photons. That means you have to have a bright light source, enough light reaching the detector, that's going to control your signal to noise if everything else is great. Okay? So that having been said, that means light throughput is what it matters. And what matters because of Beer's law is this B or path length. So they are not conducive to each other. If you increase the path length, your light intensity will decrease. That throughput will decrease. The other thing is light source intensity. If you go to variable wavelength, meaning you have a continuum source of both UV and visible, then you are basically tied to a deuterium lamp. And deuterium lamps have not improved very much over the years. So here are some typical geometries. Here you have a classical Z geometry in the macro scale. Here is a so-called U geometry, where the liquid goes like this and comes out like this. Light is going along like this. One variation of that here is that this is being used in the capillary scale. They bent this capillary into a L shape and focusing this light with a ball lens in here and hoping that this is going to reflect in there to some extent and being picked up this fiber. This is an almost impossible geometry. One tube goes in past this optical fiber all the way to the end, then the liquid comes out through here. Okay, it would be very difficult to do. One of these that have not been much used is a multi-reflection cell. When light comes, it will only work for coherent light and goes back and forth to here. Okay, the bubble cell is commercially available, often used in capillary electrophoresis, where just for the detection zone, you increase the area. Otherwise, you stick with the simplest, effective, is compromise sensitivity, but not resolution of your chromatography. Just look across the column. One other possibility is cavity-based spectroscopy. Cavity ring down is basically a physical chemistry experiment. Okay, so you have two highly reflecting mirrors. You shoot a burst of photons, and some leak into the cavity. This is where your solution is, and they attenuated by the solution passes out. Some will leak out through here, and then most of it's reflected back. So it goes back and forth. So every time, these are nanoseconds apart, every time it comes back, you have a signal. And by looking at the rate of this exponential decay, you know the absorbance. It took a while, but the inventors figured out that actually you don't have to have a pulse laser. You can do this on a continuous basis and measure the transmittance continuously and figure out the math. So people have done HPLC detectors based on the cavity principle and show that it works, but there are no cavity absorbance detectors on the market. Here's an experiment I'd like to tell you what I did. So one of the big problem with the cavity detectors are that if you have, they're always talking about 99.99% mirrors. That means if you put 10,000 photons in, only one will get in. That means if you put 100 million photons in, 10,000 will get in, and then one will come out. So that's why it's a physical chemistry experiment, that you need a very high power light source and fast detectors. What we decided to do is that let's get rid of the entrance mirror per se, not get rid of it, but make a tiny hole in it and let the light look out through it. Instead of relying on a coherent laser light, we will simply look at it reflect back. The beam will expand the moment it enters, and I'll have a broader area detector, and I can look at it. So we can, using this principle, we can amplify absorbance. This is just a plastic cuvette with those NASA blankets, shiny mylar things around with a hole in it. Okay, so this is the conventional absorbance and how much gain you're observing with uh, different number of reflectors on how many layers of the reflector I have. So it turns out they're mathematically very easily predicted. If you have 95% reflecting, then you will have a gain of about 20. Okay, and that's what you see in practice. And we have shown that you get actual signal to noise gain, actual gain in detection limit in doing so. And here is altogether different one, a photothermal detector. Here, what we're going to do is that we are going to measure the heat that's generated upon absorption of light. Okay, so imagine this is your flow cell, and I have a thermocouple here as the liquid enters, and I'm shining light through here. Light is actually coming out this way. I have a white flashlight, Walmart flashlight, and I have a little another thermocouple here, and I'm going to measure the difference in temperature between the inlet and the exit. That's my entire detector. Okay, so this is a chromatogram of, it's a white LED. There's no monochromator. These are 5 different dyes. They absorb in different colors of the region, but they absorb somewhere in that white spectrum. They all produce signals. During a gradient, you also change the composition of the solvent. The specific heat changes, that changes the background. If you subtract the background, you can get a flat chromatogram. I don't know the limits of this, but you can very easily take it to capillary scale. This is a 100 micron capillary, and you're shining a standard red laser on it. And you have the same arrangement, one before, one after, and here are some dyes. There are backscatter interferometry is something that has been greatly promoted originally by one of my colleagues at Texas Tech and now at Kansas. The this is a review, not the same. This is Jorgensen, not Jorgensen. The basically you are getting the difference in refractive index between the capillary material and causing interference, and you can detect these various analytes at various levels. This thing is ranges from 15 micromolars to 5 millimolars. So this really differs widely depending on refractive index. And so finally, I'm going to tell you the issues of measuring absorbance at the capillary scale. It's well understood technology at the pro level, and sensitivity concern at smaller scale. Things will have to absorb light for you to do this. Sugars don't work for that reason. Unless you have very bright sources of light, it's non-destructive. That's great. Difficulty in the capillary scale, it's very difficult to avoid dispersion. Let's talk about fluorescence dispersion, and we'll hopefully come to an end quickly. We all know that it absorbs light. Fluorescence is when you, instead of generating heat, you shoot out another photon of lower energy, and it detects it typically at the right angle. Okay, and how good it will be depends on how fluorescent it is. That means what is the ratio of photons emitted by photons absorbed. If you go to things like fluorescein, it's close to 95%. But one of the things here is that how the fluorescence intensity is directly proportional to the incident light intensity. Incidentally, this is the same thing in photothermal also, that how much heat you will produce, it will directly depends on how much light you're putting in. Okay, so even though it's absorbance, it's more like fluorescence that way. So you're best off measuring at 90 degrees or right angles, although sometimes people measure at um, different same direction the light came in on using a dichroic mirror, which selectively reflects one wavelength rather than the other. So fluorescence depends on how free the molecule is to move. If the molecules cannot move very well, then it cannot de-excite itself. So in lower temperature, higher viscosities, they all improve the fluorescence signal. Micellar media, you put the molecule in soap solution, it also improves. So here's a typical geometry that you have a laser diode and focusing on here, measuring the fluorescence here, and so on. This is the dichroic geometry where you have your laser being reflected by the dichroic mirror this way, and the fluorescence is, on the other hand, passes through and is detected this way. Still, this geometry, if you can do it, is better. So OTLC column that as little as 2 micron in diameter has been used with microscope optics. And finally, I am going to tell you about liquid core waveguides. So liquid core waveguides are what behaves as optical fibers. So an optical fiber behaves as an optical fiber because of total internal reflection. A optical fiber has two regions minimally, a cladding and a core. The cladding has lower refractive index than the core. So much like a fish sitting in the water, if it looks up, it basically sees the mirror because the light reflects off the lower refractive index surface and continues. So this is total internal reflection. People have tried to do this for ages now. In Japan, FUA did this in early 80s, putting carbon disulfide in glass tubes. Carbon disulfide has higher refractive index than glass, behaves as perfect optical fibers. We tried doing it in concentrated sugar or salt solution, in as well as things like zinc chloride in glass or FEP Teflon. Sugar works wonderfully, but ants find out you have sugar, believe me. The Sunoda felt that alcohol is better, but realistically, this didn't take off until DuPont in the late 90s invented Teflon AF. This polymer is completely transparent across the UV's range and has a refractive index of 1.29 to 1.31 and compared to 1.33 for water. So you have a Teflon AF tube will behave as a water-filled Teflon AF tube, behave as a liquid core waveguide. It's simple, but Teflon AF is very gas permeable. So gases are continuously coming in and out of that. So you put an alkaline solution in there, CO2 will come in and change the pH, which you may not like. So people don't usually use, and also it tends to absorb a lot of things. Here is a more common variety that is available from Polymicro, Teflon AF clad silica tube. So the total internal reflection in this case takes place at the silica Teflon AF interface. The silica is optical quality, so it has very low attenuation, even though light may go through it many times. Agilent uses nothing more than a very thin wall silica tube. Here, the the TIR takes place at the silica air interface. You have to make sure that no dust deposits on it, so you have to put it in a separate box and not open it often. So here is a situation with a simultaneous absorbance fluorescence detection. Imagine this is a liquid core waveguide. Okay, most commercial macro cells that measure absorbance, they are all liquid core waveguides. So light is going through here, goes through the liquid core waveguides to minimize loss, but not the loss to the solution. The analyte fluoresces. Fluorescence is emitted in all directions. So some of that fluorescence will proceed down, actually guided down the fiber, and you can detect it. Either detect it here, if the excitation light going this way, or you can detect the fluorescence actually and illuminate it radially. So here is a situation, for example, we have a laser diode and a capillary is sitting on top of the laser diode with a Teflon AF tube. And this is, so analyte comes through here, it's being radiated, the emitted fluorescence is carried down here to the phototube. Here is something very similar. Here you have a LED shining on that region, and the emitted fluorescence is carried down here, whereas this is transmitted, just doing absorption measurement, doing the fluorescence elicited by this, by here. So here is the same thing I'm showing you again. This is a typical flow injection type arrangement where you're looking at LED excitation, UV LED, and you detect 10 nanomolar levels of this aluminum sulfoxene, and so on and so forth. It is coumarin 30, again 50 nanomolar or something like that, and they're pretty low. So with that, it's time for transition.
Yes. Thank you very much. That was a fantastic talk. So I will just say change the slide. Oh, I'll share for us then. Thank you very much again. That was a wonderful talk. And with that, I'll be taking it over to discuss a little bit about mass spec. For the sake of time, we're going to keep it very brief and very broad. And if anyone does have any questions, I would ask you to please put those in the chat now. And if we don't get to them, we will at the end. So a little bit about mass spectrometry, just a very broad overview. In mass spectrometry, you take ionized gases and typically manipulate them through an electromagnetic field. And based on how they separate, you can identify the charge to mass ratio, or the M/Z. And this allows you to obtain information and often very high sensitivities of these mass analytes. Now, how you actually move them through the system and perform those separations, there's a couple of common techniques. So first and foremost, this is sort of the low-end system, the quadrupole mass spectrometer. This is a simple system in which you have a quadrupole, so two sets of rods, and you apply a frequency between them. And based on the changing of the voltage and the frequency, you're able to separate these compounds based on their specific mass to charge ratio when they enter the detector. This is very great. It can provide you an overall mass, but it does struggle to provide specific structural insights. Because of that, they have introduced something called the triple quadrupole mass spectrometer. This is a furthering of that mass spec system. So you take your initial single quadrupole to filter out mass to charge ratios. You then have your second, or Q2 region, where they have a collision cell which fragments your molecule. And based on that fragmentation pattern, you can analyze it with a second, or in this case, Q3 mass filter, which will give you the M/Zs of those fragmented sections. This offers higher sensitivity typically, and it also offers some structural elucidation based on the fragmentation patterns. It does often cost more and requires a larger benchtop footprint, so it is a pro and con there. Another common one, and this is a really cool one, is a time of flight mass spectrometer. Here, ions are introduced into the system and effectively shot at something called a reflectron, which turns their momentum around or turns their velocity around. And based on their actual time of flight or the time they spend in this flight tube, you can actually determine their mass to charge ratio. And so it's another way of identifying it. In this case, rather than using alternating currents, you can base it off of their ability to move through that flight tube. Next, we have the Orbitrap mass spectrometer. This is a much more high-powered and very interesting system in which the ions are actually sent to rotate around a central core. And as they rotate around that core, they generate an electrical signal, which when you perform a Fourier transformation on, you can elucidate the mass spectrum of whatever ions are in your chamber at any given time. This one's very high-end. It's very fast acquisition rates, and it does offer very high mass resolution. And so it's another option. Now, as for getting your samples into these ionized gas phases, I want to focus on two key techniques. There are a number of them, but we're skipping them due to time. So one of the common ones is APCI, or atmospheric pressure chemical ionization. In this case, the eluent or the effluent of your LC is nebulized by a stream of heated gas, which evaporates the solvent, getting everything into the gas phase, and then a coronal discharge needle imparts a charge onto the systems, or onto your analytes before sending them to the mass spec. Now, this is great at ionizing nonpolar, polar, or ionic compounds. You don't see many impacts from your matrix effects, and it can operate at very high flow rates. However, as we're going to get into, you typically don't see the same sensitivity gains that you would if you were working with something on the capillary scale at these lower flow rates. So when you're working with electrospray ionization, which is in this case one of the more common forms, and in fact often used with these lower flow rates, in electrospray ionization, you apply a high voltage to a capillary. There, the electrical force breaks the surface tension of the liquid, resulting in a spray of very small droplets that contain both your analyte and your solvent. As these droplets start to evaporate, they actually concentrate the charges in, which eventually again overcomes surface tension and explodes the particles into even smaller droplets. Now, when you reduce your flow rate, you often reduce the inner diameter of your tubing, and you're able to produce smaller and smaller droplets, which ionize better, and they often produce better sensitivities. This does work on very large polar and ionic compounds, but it does struggle with nonpolar compounds, which is a drawback of the technique. Now, when working at these lower flow rates, you often see very significant sensitivity increases. There was a sensitivity increase reported in the literature of anywhere between 4 and 10 times higher sensitivity. Again, it will depend on the source and the analyte, but that's just one literature value. Comparing that to the Axcend system itself, and I'm running out of time here, so I'm going quicker, we were able to see similar 10-fold sensitivity increases compared to analytical scale when running a peptide system. Moving forward to the optical detection, Axcend also offers our own optical detectors. The first is on-capillary detection with our fixed column cartridge. Here we're performing that detection directly at the outlet of our system. And so because of that, we limit the amount of dispersion, but we're also limited by the inner diameter of the column. And so because of that, we often see lower sensitivities. Because of that, we implemented a Z flow cell based detector. In this case, it has a 1.2 millimeter path length, which does increase the sensitivity, and we'll show a side-by-side comparison for that. It does introduce more dispersion because it isn't directly at the column outlet. However, the sensitivity gains from the flow cell often outweigh the dispersion concerns. So just to quantify that, and then I'll wrap up, our on-capillary detection versus our flow cell detection, we compared it in a direct head-to-head separation of alkylphenones, and we predicted about an 8-fold increase based on the we'll call it Beer's law, although we've learned that that is not the proper terminology. But based on that, we would predict an 8-fold sensitivity increase. We saw a 7.8-fold sensitivity increase. So some of that dispersion was coming into play. However, we still did see those gains. Now, just in wrapping up, if you are interested in learning more about Capillary LC, there is a public capillary innovation forum. There are a lot of experts there, and if you're interested in learning more or becoming active in the community, I really would urge you to join. And finally, if anyone did have any questions or would like to learn anything more, the Axcend website is there for you. I'd be happy to answer any questions. We are over time, so we will cut it short. But thank you all very much. And I'd like to again thank Dr. Dasgupta for taking his time to show us some really phenomenal chemical detection systems. That was an excellent talk. And with that, thank you very much.